16 second half support and movement for the preparation of PPSC lecturer and subject specialist zoology and biology. In the first part of this chapter, we have discussed support in plants up to deformities of skeleton, but in this second part, we shall discuss from types of muscles up to locomotion in vertebrates. Now, that is the second most technical chapter in biology after respiration. Allah says in Quran that indeed Allah will not change the condition of people until they change what is in themselves. Surah Rad, verse 13. Let's start. Muscles. Earliest form of muscles to evolve was smooth muscles. They are actually found throughout animal kingdom in all the animals. That's why it is suggested that they are the earliest form. Smooth muscle cells are long, spindle shaped with single nucleus. That spindle shape is important for you to focus. They do not have striations and not controlled voluntarily. They are actually involuntary. Blood vessels and digestive tract are mostly made up of smooth muscles. Please remember. Cardiac muscles are chains of single cell, each with own nucleus. So that is the chains of the cells in cardiac muscles or heart muscles. They are striated and involuntary as well with the branches. So the only branched muscles or cells are actually cardiac muscles. Skeletal muscles are voluntary muscles. They are under our control. They are striated due to alternate light and dark bands which are obviously or clearly seen biceps and triceps. Tendons are bundles of collagen. They are non-elastic and they attach muscles to bones. Skeletal muscle fiber. Each muscle is made up of muscle bundles which are consisted of cells or fibers. So when we talk about a muscle fiber, we are actually talking about a muscle cell. Each cell or fiber have multiple oval nuclei just beneath sarcolemma. Sarcolemma is the cell membrane or plasma membrane of a muscle cell. Please remember. These are huge cells with diameter of 10 to 100 micrometer, muscle cells or muscle fibers. Sarcoplasm differ from cytoplasm only in having large glycogen storage and the myoglobin, which these two things are not present in a typical cytoplasm. Each cell or fiber has large number of myofibrils extending entire length parallelly. Each myofibril is around 1 to 2 micrometer in diameter. Each myofibril is consisted of smaller contractile units called sarcomere. Each sarcomere has dark and light bands. Now, each dark band is called A band while each light band is called I band. Now your attention must be deeper and please follow each word in with your mind. A for anisotropic mean it can polarize visible light, while I for isotropic mean it cannot polarize the visible light. That's why the word A and I are used. Lighter stripe in midsection of A band is called H zone. H from hele, which mean bright. So that is also called as bright zone. Now, H zone is bisected at right angle by a dark line called M line. Midline of I band is called Z line and that Z from Zwischen mean in between. Sarcomere is the region of myofibril between two successive Z lines. Sarcomere is the smallest contractile unit of muscle fiber and that is important point, please focus. Myofibril contain myofilaments. Now in this diagram, you observe that the upper part is the real image of a muscle while in the lower part it is the diagrammatic scheme or schematic diagram to explain the upper structure. So in that you can say that these purple colors bands are actually A band and they are myosin, while the light colors or orange or green lines are representing I band on either side. Similarly, you can observe in both I band a zigzag line which is actually called as a Z line, as well as an empty portion between the two stripes of A band which is called as H zone actually, and that H zone is bisected by the M line at 90 degree So that is the basic scheme of a sarcoplasm or a muscle fiber. Now, infrastructure of myofilament. Central thick filament, which is made up of myosin, extends in entire A band as we have observed in the diagram. Now the thin filament is made from actin, extends across I band and partly into A band. Each myosin, almost 16 nanometer molecule, has tail terminating into two globular heads. These two globular heads are actually very important for the initiation of contraction. Myosin tail consists of two long polypeptide chains coiled together. Terminating heads of these chains called cross bridges because they link both thin and thick filaments during contraction. Each thin filament is 7 to 8 nanometer thick and is composed of actin. These actin molecules are arranged in two chains twisted around each other. These two chains are further twisted around by two strands of tropomyosin. Now that is very important MCQ. The third protein in the thin filament is troponin, which is 3 polypeptide complex itself. One polypeptide of troponin binds to actin, second to myosin, and third to calcium ion. Please remember these MCQs are very boring but they are very important. Now sliding filament model. Each myosin is surrounded by 6 actin filaments on each end. Please remember this fact is very important. When muscle fiber contract, shifting occurs in thin and thick filaments across each other. I band reduce in size and Z line come closer. Sliding filament was hypothesized actually by Huxley and A.F. Huxley in 1954. According to this model, the thin filaments slide past the thick one so that actin and myosin filaments overlap to a greater degree. During contraction, Z line come close, I band shorten and H zone disappear. These three keywords are actually very important for MCQs. Cross bridges of thick filament attach with binding sites of actin filament. Now these cross bridges then contract to pull the actin towards center of sarcomere, and that's the sliding filament model. Now how these bridges are controlled, that's another important thing to understand. At rest, tropomyosin covers the sides on actin chain, so they cannot attach with the bridges. During contraction, calcium ions bind with troponin to cause slight movement. This slight movement displaces tropomyosin and sides are exposed. Then myosin head attach with actin filament followed by ATP hydrolysis. This ATP is provided by large number of mitochondria present in the muscle cells. Actually, ATP breaks the links between myosin and actin, or in other words, we can say it breaks the cross bridges. After death, ATP amount falls in body, so bridges cannot be broken or cannot be separated, and that is the reason of body stiffening after death, which is called rigor mortis. Actin-myosin interaction. Contraction is initiated at neuromuscular junction when impulse arrives or message arrives. Complete motor unit contract simultaneously in response to action potential. T-tubules are extension of sarcolemma which are continuous extracellularly as well. Thousands of such T-tubules in a single cell are called T-system. T-system encircle the myofibril at Z-line or AI band junction. Sarcoplasmic reticulum form triads with T-tubule at intervals along myofibril. Nerve impulse is carried to sarcoplasmic reticulum through T-tubule, that is not directly to sarcoplasmic reticulum. Calcium gates of sarcoplasmic open and release calcium ions in cytosol, which then bind with troponin of thin filament to expose binding sites for cross bridges. Now muscle contraction is all or none response. Degree of contraction depends on number of fibrils which are active. So sarcoplasmic reticulum do not have ribosome, and that is the main difference from the typical endoplasmic reticulum. Sarcoplasmic reticulum do not have ribosomes at all. Now energy and fatigue, the basic concepts. Stored glycogen in a cell is converted to glucose, and then that glucose is converted to ATP for muscle contraction aerobically. Creatine phosphate is another energy storing substance, and the third source of energy in the muscles is anaerobic breakdown of glucose into lactic acid. Now, accumulation of lactic acid causes muscle fatigue. At rest, only 20% of lactic acid is broken aerobically, and that energy is used to change remaining 80% lactic acid back into glucose when we are at rest after hard exercise. Basic cause of muscle fatigue is lack of ATP, due to which detachment of cross bridges halt or stop, and they remain attached to cause prolonged contraction. Accumulation of lactic acid causes pH of muscles to drop or low toward acidic, and hence ache or pain and extreme fatigue occurs when lactic acid breaks glucose. Now, how exercise affects and what are tetany and cramps? Exercise result in increase in size of muscles, strength and fatigue resistance. Capillaries surrounding muscles increase, as well as mitochondria increase in the cells when a person do exercise consistently and continuously. Myoglobin is synthesized in muscle fibers. Complete immobilization of muscles result into atrophy, or we do not move at all, then atrophy results. Low calcium in blood cause tetany disease, which increase excitability and loss of sensation of neurons. Please remember that tetany is related with the excitability and loss of sensation of neurons. Muscle twitches or convulsions occur, and if untreated, tetany untreated leads to spasm of larynx, respiratory paralysis, and ultimately death. Now, tetanic contraction of entire muscle is called cramp. Cramps reflect low blood sugar level, electrolyte depletion, dehydration, and irritability of spinal cords and neurons. These are actually symptoms of the cramps. Fundamentals of skeletal movement, some definitions. Origin, insertion, and belly are three parts of a muscle. Origin is stationary. Insertion moves the bone while belly contracts in between. Ligaments attach bone to bone and are elastic, while tendons are non-elastic and attach muscles to bones. Clostridium tetani, that is a bacterium, causes acute infectious disease, the tetanus. Tetanus is fatal due to respiratory failure, and up to 40% of the patient die if that disease occurred. There are 650 muscles in human body in total. Each bony lever is moved by an antagonistic muscle pair or alternate contractions. Biceps brachii muscle arises from two heads of scapula and is inserted in the radius bone, while brachialis muscle is inserted in ulna bone of elbow. These two are very important facts for you to remember for MCQs, please. Now, locomotion in invertebrates. We shall go very rapidly and quickly, only discussing the important things. When flagellum moves forward, euglena does not move backward. That is the distinction. Flagellum is at interior end and pulls the body forward. Waves in spiral fashion pass from base of flagellum to tip, which cause rotation. Active contractile myonemes help to change direction in euglena. Please remember, these four facts are very, very important and the filtrate of the reading. In paramecium, ciliary wave starts at anterior end and progress to posterior. So, not all the cilia beat at a time in a single direction, but one by one or step by step. Cilia are extensions of cell membrane with diameter of 0.1 to 0.5 micron. Each cilia has 9 pairs of fibrils at periphery or corners and 2 fibrils at center longitudinally. In 1955, Bradford suggested movement of cilia by sliding off double fibrils in two groups. That is, 5 out of 9 contract to bend cilia in effective stroke, while 4 out of 9 contract to straighten it in a recovery stroke. So that is the difference between a effective stroke and recovery stroke. Enzymes are present in cilia to hydrolyze ATP to provide energy. So that question can be asked that from where the cilia get energy. Now, jellyfish moves by jet propulsion mechanism and the body is called bell. Water is filled in the body, then the circular muscles contract, push the water outside, and jellyfish moves forward. Accordion-like movement is shown by earthworm due to setae and muscles. First, earthworm become long and thin. Please remember that is important MCQ and has asked in the past papers that the first step of the earthworm locomotion is long and thin body. Anterior setae anchor and hold, then longitudinal muscles contract. So in the first phase, longitudinal muscles contract and body shorten, pulling it forward. Then posterior setae anchor and circular muscles contract. So that is the method of locomotion in earthworm. Cockroach can swift walk and fly. Both modes of locomotion are present in the cockroach. Foreleg and hind leg pull and push on one side with opposite middle leg acting as props. So these three legs at a time are working together, two on one side and the middle on the other side. Same is repeated for other three legs. Posterior pair of wings brings about flight in cockroach. Only posterior pair, not anterior. Snail moves or crawl by muscular foot. Starfish moves with tube feet and arms also help in swimming. So these are the salient features which I have drawn from the text. Now, vertebrate locomotion fishes. Streamlined body, only fins project outside, nothing else. Dermal denticles in cartilaginous fishes and scales in bony fishes are made slimy from mucus or oil to reduce friction. That is the most major adaptations to locomote in the water. Dorsal and ventral fins are unpaired, which stabilize the body. Pectoral and pelvic paired fins steer and balance the body, and caudal or tail fins propel body forward. These are the different functions of these fins, and your attention is needed to understand and remember them. Amphibians. They have journal build like fishes, but they can wriggle along their belly with the help of segmentally arranged muscles, which is just like the swimming on land. Other amphibians raise their body on legs, which act as movable levers. Swimming and jumping is specialized in aneurons due to extensor thrust of limbs. Frogs and toads walks and hops due to strong hind limbs. Hops mean jumping. Now, reptiles skeleton is inherited from ancient amphibians, which is highly ossified or bony. Atlas and axis vertebrae, which are actually first and second vertebrae of cervical region. So cervical region is present in the reptiles. Atlas and axis vertebrae of cervical region provide freedom of movement. Axis is modified for rotational movement. That is the key point here. Snake ribs are connected with belly scales to aid in locomotion. Many historic reptiles were bipedal, that is, they used to walk on two legs, that is hind legs. They had narrow pelvis and long tail for balance. So the function of the tail was actually balancing the body. This bipedalism freed four appendages for prey capture and flight. Birds. Skeleton modified for flight with large air spaces in bone. That's why the bones are very light in weight. Sternum is modified to form keel for attachment of muscles. During gliding, wings act as aerofoils. Angle of wing with air provide lift. In active flight, flapping of wings lift the bird up due to low pressure on upside. Mammals. Plantigrade are those type of locomotion or animals or mammals which walk on soles with palm, wrist and digits all resting on ground, such as monkeys, apes, man and bears. So we are actually plantigrade. Digitigrade walk on digits only, run faster than plantigrade, and first digit is reduced or completely lost, for example rabbit and rodents. Unguligrade walk on tips of toes modified into hoof like deer, goats, or donkey or horse, etc. It is most swift type of locomotion. So these are the three basic types of locomotion in mammals, and they are very important for you to remember. Now S-band locomotion occurs in fishes which pass anterior to posterior. So S, as the shape of S suggests, that is the actually movement of the body in the water. That's why that is called S-band locomotion. Tetrapod pelvic girdle is united to sacral region of vertebral column. Please remember, acetabulum is a depression between ilium, ischium and pubis for femur attachment in the pelvic region or pelvic girdle. Flight has evolved in pterodactyls, birds and bats only. So that is it for this second half. Inshallah, the next lecture will be of chapter 17, first half. That's also a very long and tough chapter, but we shall divide it into portions with easy facts, understanding and concepts with diagrams as well. To watch other chapters, you can check the playlist or description of this video. Thank you so much for staying with me. Stay